Hey, hi everybody. Happy Friday. Welcome back to this week's Single Cell and Exploration Club, where we usually use a case study to demonstrate some of the features on our platforms. But for today, it's very exciting because we're actually introducing the version 5 of Talk2Data. So we've released a lot more studies, a lot more cells. As you can see here, we have over 200 million human cells, over 1,000 cell types, and over 3,600 data sets annotated for Talk2Data version 5. And so before we jump into the agenda for today, we're going to talk a little bit about our talk from last week. So if you can switch the slide, please.
Okay, let me just switch the slide. Yeah.
Okay, thank you. So from last week, we dived into this data set, the Tahoe 100 million data set, and we've garnered a lot of interest from people who watched our last talk. So from here, this is the preprint paper in which you're able to see the data set and see how the authors have analyzed it themselves. So within this data set, there were 50 cancer cell lines, 1,100 drug dose perturbations, and they also use this data set for AI model training as well. And this data set is available on Talk2Data, so Hazel will show you where you can find it on Talk2Data.
Oh, yes. Thanks, Julie, for the introduction. And this is Tahoe 100 million. And let me just quickly guide you how you can access it on the Talk2Data.
Oh, it looks like we lost Hazel for a little bit. Maybe give her a couple seconds to come back online. Otherwise, I can try sharing my screen as well. Sorry, everyone. Some slight technical difficulties. All right. I'll try opening up the data set on my side. Give me 1 second. Okay. Sorry, everyone. I'm also having trouble sharing my screen at this moment. Give me 1 second, please. Sorry, everybody. I'm having a lot of trouble sharing my screen as well. I may have to exit and restart the Zoom. So if everyone can just sit tight for 2 minutes, we'll be right back. Okay, thank you, everyone.
Hi, everybody again. I'm really sorry. My internet connection is just suddenly dropped. But let me share with the screen again and we can start. Okay. Okay, so here we are. Here we
Okay.
are. Yeah. Oh, hi, Julie.
Hi, we're both back. So sorry about that.
Yeah, I'm really sorry. Okay, so we were in the part where I tried to show you how to access the Tahoe 100 million. So you can quickly go, okay? Like from the Talk2Data, you can click go now, and it will navigate you to this interface. By default, the database that you are in right now is the database version 5. As you can see, the Bioturing default project. So how can you know? This is like there is a small setting button on top of the screen right here. And click that, and it will show you that right now you're in the database version 5. And if you want to switch to Tahoe 100 million, just simply change to database version Tahoe 100 million and save the setting. Okay. And like that, you will be navigated to the Tahoe 100 million. Okay. Now let me go back to the slide where Julie can start, showing you what we will present today.
Yeah, so for today's agenda, we're going to go through two sections. So in the first section, we are going to cover how our data team here curates the data. So usually the process of how this happens is one of our users will send in a request for a data set curation, and then it'll go through a couple processes to standardize the ontologies, some QC steps as well. And so Hazel will be able to cover that. And in the next section, we'll talk about what's new in the database version 5 and some of these standardized ontologies that you can see in version 5.
Okay. Okay, now let's start with how did we curate the data. So, so this data curation process, a summary. So as you can see, within the data within the curation process, we have 4 main steps, right? Okay, so the first steps would be data request. So as Julie just mentioned, our data source is from publicly available data sets. And the why we know these data sets, it can come from two sources. First, it can come from our users' request. So if you are like a Bioturing users, you can freely request for any publicly available data set. Just send us a request and we will curate that one if that's available. And beside from that, we also collect newly published paper on a daily basis. So we have like a data team to search, to browse like the internet and Google Scholar to find newly published single cell RNA-seq data. And then after we got the data, we could check the data to see whether it comes in full, whether we have like enough the expression matrix and the metadata within to make sure that we can start curating the data set. After data check, we will go to annotate the cell. Actually, we don't do annotate the cell ourselves. We have usually in the publicly available data, we have the authors who provide us the annotation, and we'll go into detail of this part later. And after everything, after the data and the annotation, we'll have 2 rounds of quality controls, okay? So now we will go deeper into specific steps and the criteria involved in each of these stages. So first of all, it's a data request, right? Now this show you like a closer look of how this part is, like how the curation process is initiated. And as mentioned, requests can come through our request management page. As you can see right here, if you access within the Bioturing ecosystem, you can click onto the request management button, okay? And this is like a central hub for submitting and managing data requests. And within the request management page, you just need to click on the data, like the request data button here, and it will lead you to a form, right? So this is like some example data that I already fill in. So you can put in like the paper title and also the link, but usually the title is enough for us to check. Okay, and after that, you can submit. There's a little submit button here. Okay, you can submit submit your request and our data team will receive. And usually the turnaround time is within 2 weeks, right? Beside that, beside this request management page, you can also send us like an email to support@biotrain.com to ask for some specific data, or let's say if you need something like really urgent, just please let us know so we can prioritize those requests. Okay. And that next we go to the data check. So as you can see in this flow chart right here, this stage involve like an evaluation of the submitted data. So we begin by checking if the essential data files are available, right? So when we say full data, as you can see here, when we say full data, we mean the data set that the authors provide come with like the expression matrix, come with full metadata information about how many samples are there and also the clinical information, if any. And also, usually we also need the authors to provide us with the number of cells after filtering, so the QC parameter from the authors. Okay, and here, if the full data is available, we will move on to the next step, which is the annotation check. In here, I just want to head up for you guys that ideally, we prefer to receive raw count matrices as indicated by the asterisk here over already normalized data, right? And okay, right here. And in case that there is no data available online, so usually within the paper, there will be the link to the data deposit place. But if we cannot find that, we will try to contact the author and to say like, if there any data available, and if there is no data available, so unfortunately, these data sets will not pass our QC, and we will let you know that, okay, so we cannot retrieve the data for this data set, right? Okay, so after this process, we move on to the annotation check, okay? So within the annotation check, so as you may know that this is one of the very popular questions that we receive from users that, so where do the annotation come from, right? Where do the annotation come from? So actually this flowchart can provide quite like full information about that part. So first, we will check whether the cell type annotation were provided by the authors or like, by the authors of the paper. And if the annotation are provided, we will then evaluate if they are, first of all, within the object that the authors provided us, if they have already provided, individual cell barcode alongside with their own annotation. So we will just submit the object in full. Beside that, we will also check, like double check the annotation using the marker genes that the authors provide within the paper, like as you can see here, for the useful marker, right? Okay, and if the process going well, like the annotations are there and after we double check with the markers that the author provide, everything match, then the paper will pass QC1, okay? And another thing within this curation process is that when the authors provide the annotations, so they are authors annotation, there will be a lot of, of iteration between different authors. If like a little bit differences, like let's say B cell, but in different vocabularies, we will standardize those terms and turn them into like a standardized vocabs within the Talk2Data. Okay. However, oh, I'm sorry, I see one like Q&A, one question here.
Oh, yes. So
No.
this user asked, so if anyone who has not yet subscribed to bbrowser X, if you're interested, you can always send an email to support@bioturn.com. All you need to provide is your email address, and one of our team members can follow up with an appropriate quote based on your research needs.
Okay. Yeah, thank you, Julie. And now we move on to the next part. If the annotations were not provided, then we'll contact the authors again to ask them, okay, is there any way that they can provide us with like detailed cell barcode with annotation? Okay, like that. But sometimes there is no annotation provided. In that case, I want to highlight this part because even when the authors did not provide the annotation, there still will be annotations from the paper. There's just no detailed data for that. In that case, we will use macrogenes from literature and also the macrogenes that the author state in the paper to annotate the cells again based on what the author state in the paper. And after the annotation process, if we got like good annotation. So what is good annotation? Good annotation means like the annotation come, like let's say the clusters of each cell types assemble quite the same. Let's say that like 90 to 95% similar with what author state in the paper, then we will use that one. We will use that annotation and it will pass the quality control one, the first round quality control. Otherwise, if we cannot reproduce the annotation that the authors state in the paper, in that case, unfortunately, this data set will not pass the QC. And I want to make sure that in this part, the reason why we do not dispose the data set if we cannot, receive the detailed annotation is that we understand single cell data is very, very precious. One data set is a very precious thing to us. So we want to use as much of it as possible. Okay. And yes, that's about the annotation check. After the annotation check, the data set is now past QC1. We will then move on to the QC2. So after the complete of annotation and data and submit the data set into internal testing, we will perform another round of quality control. Within this round of quality control, we will check the completeness of data again. Why? Because usually the annotation process after the first QC, like quite some time later, let's say a few months or a year later, we will check again. Because sometimes in the first round, the paper is preprint. And a year ago, the paper is now fully published. Maybe the title are changed, the data deposit plate is changed, and even sometimes in rare cases, the number of samples change. The author, they can increase the number of samples. In that case, we will take the data set and bring it back to the first place to, to check the data again and re-annotate everything again, okay? The second thing we will do within the second round quality control is to check the filtering parameters. So we check to see whether the filtering parameters are applied correctly or not. Usually, let's say 90% of the time, the filtering parameters are applied following the original paper. In the paper, under the method part, usually there will be the QC params of each data set, and we will apply the same. However, in some case, if the parameters are not provided, we will do it like this. So if authors provide the cell numbers, usually the cell numbers are attached with the UMAP. In that case, we filter based on the distribution of number of genes and transcripts to approximately reach that number of cells. And if the authors do not provide the cell numbers, this is a hard case. In this case, we will slightly filter the outlier cells based on the distribution of number of genes and transcripts. And then after the cell type annotation, we will continue filter cells by removing clusters that do not have any enriched macrogenes. And this principle is applied similarly for the the mitogenes. And in here, because I mentioned about the distribution of genes and transcript, I also want to mention that within Talk2Data, for each data set, you can view this threshold. So let me show you how you can check the threshold like this. As you can see, if you open any data set within Talk2Data, you will navigate it to the single cell exploration UI, right? The interface. And within the metadata list, you will find 3 metadata of number of genes, number of mRNA transcript, and the percentage of mitogenes. Within each of these metadata, you will be able to see the threshold that we apply for individual data set. And I think this is a good chance. Why? Because first of all, you can double check with the original paper, or you can also filter the data even more. Like, let's say you can see that the number of transcript a little bit too low. So you can open the metadata and you can filter it out. Let me show you how can you do it. So this is one data set, for example, and this is the number of gene. Okay, let me just zoom in a little bit. What you can do is that you can set the threshold of your own by dragging along the distribution plot. Okay, by dragging along the distribution plot, you can reselect the cells based on the number of genes or number of transcripts. Right now, let's say that I am selecting around 250,000 cells based on the cutoff level of 600 genes per cell, right? Okay, now let me go back. Let me go back here. And then that's everything of our curation process. So within this, within this section, we will also share with you like a short version of our curation pipeline, where you can, this is just like a PDF file. So you can have a quick view of how we actually curate the data. And now we'll move on to the next part, where Julie will show you some of the new characteristics of Talk2Data database version 5. Okay.
Yeah, so in this section, I basically want to give you guys an overview of all of these standardized ontology terms that we have. So in the next slide, you'll be able to see that overview where we have over 200 million cells. These are human cells. We do have a database for mouse as well, but we will be releasing that sometime in the future. So if you can move to the next slide. Okay. Yeah, so we have more than 200 million human cells, over 3,600 data sets, 100,000 or more cell types, over 300 treatments, over 500 detailed conditions. So these are either diseased or healthy, and over 400 tissue types as well. And one of the greatest benefits to this single cell database and these standardized ontologies is that we're able to integrate it into our other apps. SmartBulk, which analyzes bulk RNA sequencing data, bBrowser X Pro, which analyzes your own internal single cell sequencing data, as well as SpatialX, which analyzes spatial data. So utilizing these ontology terms, you're able to integrate these apps and connect them in this way. So moving on to some of the statistics that we have for the types of data sets that we include. So for this version, we shifted towards more immuno-related studies. In our previous version, we had a lot of data curation for cancer-related studies, but for this new version, we wanted to shift that towards more inflammatory diseases, for example. So you can see some of the top categories here include dermatitis, gastroenteritis, hepatitis, and so on. And so in total, we have 267 studies relating to inflammatory disease, and also, again, over 20 million cells relating to inflammatory disease. Okay. And then in this next slide, similarly, we have the immune system disorder studies. So studies related to immune system cancer, autoimmune disease, leukocyte disorder, bone marrow disorder, and so on. Here we have 471 studies related to immune system disorders. Okay. So in this next ontology term, we'll talk about some of these cell types that we cover. A lot of the cell types relate to immune cells. You can see that through B cells, NK cells, T cells, and macrophages. But what's interesting here is that you see the spike of cells in neurons. So neurons do constitute a very large proportion of the cell types in Talk2Data as well. So if you are a scientist interested in researching immune cells or neurons, you can definitely find a lot of studies on Talk2Data relating to those.
Okay.
Okay. So moreover, we also standardize the ontology terms for the sampling site as well. So this is very relevant to those of you who are studying cancer so that you can see where the sample is taken from. For example, is it taken from healthy tissue, from primary tumor site, from diseased tissue? We also have an adjacent normal tissue as well as the site of metastasis.
Mhm.
Okay. And then this one I also find really interesting as well, where we standardize the treatment ontology. So here you'll be able to find a lot of cancer targeting treatments. For example, pembrolizumab, nivolumab, and anti-PD-1 all inhibit PD-1. So if you're interested in this type of research, for example, breast cancer or anything relating to the PD-1 gene, you'll definitely be able to find treatments and studies relating to these treatments targeting PD-1 in Talk2Data. Okay. Moreover, we also include the treatment status so that it's easier for you to find which studies have a treatment, which versus ones that don't. And here you're able to find over 400 studies that do include some sort of treatment. So you can look further into those studies to see what kind of treatment that they offer. Next, again, relating to treatment, you'll also be able to view the treatment response. And we standardize that in very simple terms. Either the sample or the patient responded or did not respond or had partial response or remission if it was relating to cancer or if they were in relapse, for example. So these are all very useful terms for you to do some time point analysis. Okay, next here. So some people might be interested in the suspension type as well. So the Talk2Data database not only includes single cell RNA sequencing data, but it also includes single nuclei sequencing data. Excuse the caption on the right, but you'll be able to see in the figure, the labels. We do have a large collection of single cell, but we are starting a collection of single nuclei data as well.
Yep.
Okay, so here you'll be able to see the quantification or the pipeline that was used to generate the data. So most of these studies on Talk2Data, they come from the Cell Ranger pipeline. So you can see here, we represent Cell Rangers version 1 through 7. But we also include some other quantification methods such as BD Rhapsody or DropSeq. Okay, this one's pretty simple. You'll be able to see the different genders that we have in the database. We do have similar representation across male and female, and near equal representation of the studies between male and female samples. Okay, here you'll be able to see the developmental stages as well. So how old the patients are within each of the studies. For some of the larger proportion of studies, most of them are adults. And within those adults, a lot of them are aged 60 to 69. So many studies in the database encompass adults 50 years or older. Okay, here you can also see the assay that was used. So most of these studies are sequenced by 10x Genomics. It's definitely a very, very popular sequencing platform among scientists, but we do also have other technologies as well, such as SmartSeq, DropSeq, BD Rhapsody, and so on.
Okay, and I think that's like some of the statistic of like to show you the characteristics of Talk2Data beta version, I'm sorry, the Talk2Data version 5, our new database. And I just want to quickly show you that along with like the new database, we also have new features and new UI, right? So as you, let me move to the Talk2Data real quick. As you can see right now, the interface is a little bit, different from that of the previous version. And so we already put some use case so that you can quickly see how the platform can be used. For gene search, let's say for gene search, what you can do is you can query the gene across the whole database. And beside the whole database, you can view the gene expression across all the metadata information that Julie just mentioned. Let's say that you want to view like gene expression in different conditions, in different tissues, and in different cell types. So that's one thing you can do because we want you to utilize the whole database so that, yes, now you can work and perform analysis with up to 200 million cells, and there's no limitation on this. Let me go back and I will quickly show you some new features. So this is what we call a project, and this is the new thing in the new Talk2Data. So a project is actually a custom atlas, and a custom atlas here, this is like a set. So usually we define the custom atlas as a set of studies, right? Different study, you combine different studies into, into one custom data set, and you analyze them together. So in previous Talk2Data, we understand that the limitation on the cell number for custom atlas may be a huge disadvantage of exploration, and we don't want that to happen again. So yes, in the new Talk2Data right now, you can either work with the whole database for your gene query and for the composition analysis, so you can understand, like, let's say the cell composition in different type of cancer or different kind of diseases. Or you can select the studies of your interest, as I showed the example here, and then you can combine them together as many as you want. So, okay, that's a project. And what can you do with a project? What can you do? So after you, I'm sorry for this part. I think there's a little bit of, let me just quickly show you this one. Okay. So after you creating a project, you can query the gene across the whole project. You can view the gene expression, as I just mentioned, like in here, for example, view the CD3 expression across different cell type. After that, you can do next is you can perform the composition analysis to compare the gene expression between multiple cell populations, right? Let's say you have
Before
a
we move on,
population
Hazel,
of,
we do have a question in the Q&A. So regarding the last slide, so you showed the expression of CD3D, I believe. So this user is interested in seeing if you can distinguish between single cell and single nucleus. So she wants to know how to search for studies that are single nucleus versus single cell, or how to filter it on Talk2Data.
oh, sure, Maria. Let me just try to quickly show you. In here, I think I will need to move back to the database version 5 first, because I was originally in Tahoe 100 million. Okay. So here I am. Let me quickly go here. So for to select studies based on either single nucleus or single cell, okay, what you can do is that, so let's say that you would just want to work with single, okay, let's see, single nucleus, right? You want to use studies that use single nucleus only, you can go to the study tab right? You can go to the study tab right here, and you can open the search. Let me delete this one. And in here, we have like a sampling size metadata, suspension type metadata, where you can just click and select, okay. You can select the nucleus. So we have around 400 studies about nucleus and apply the search, right? And I think also this way can have us to answer the question from Sandrine as well, like how you can select my project or custom atlas from the libraries, okay? After I create the project, I will then show you, right? So this is like all the study using the nucleus in the database. I can select everything, 400 studies, and create a project. This is like study use nucleus, all right? Create the project, okay? Create the project, okay? Like that. After that, you can see that right now, you are under the project of studies use nucleus, okay? Okay, Maria, so I hope that this can help you to answer your question. And now I will move to Sandrine. So after you create a project, you can go to My Libraries, okay? And within the My Libraries, you will see every project that you have created. And just simply click onto the project name. For example, here I have like an inflammatory disease project. I can click onto that and it will bring me to the project. Okay. Okay. And now let me go back to the slide. And also, okay. I think it's a good opportunity also because we're already in the project. We're already in the project. So as you can see, first you can create the gene across the project and you can perform different types of downstream analysis. You can see different analysis on the right side of the screen here. First, you can run the composition analysis. You can run the correlation analysis, the marker gene, and the DGE. Okay, here are the place where you can find the tune. And now I will go back to the slide to show you a little bit in detail. For the composition analysis, you can compare the gene expression between different subpopulations. We call them the experiments, but they're actually different subpopulation. Or you can select different cell types and compare the cell composition between different conditions as well. For the correlation analysis, you can do two things, right? You can have a gene of interest and you can input the gene into the system and ask us what are the top co-expressed genes with your gene of interest in a specific condition. Let's say that what are the top co-expressed genes with PDCD1 in lung cancer and that kind of thing. Or the second thing, what you can do as I show on the screen here is that you have a list of genes and you can run a correlation analysis using the Pearson score or the Jacquard score to find the pairwise correlations between, okay. Oh, I'm sorry. Okay. Okay, and find the correlation of each pair within the gene list in a specific condition. In here, I'm working with tumor enterocyte versus non-tumor enterocyte, for example. Okay. For the marker genes, it's just as in the name, you have like a cell type of interest and you want to find some genes to distinguish that cell type from others. Let's say in here, I'm working with kidney and I want to find the genes that can distinguish the podocyte, one cell type in the kidney with other cell in the kidney. And I just need to select podocyte as my cell of interest. And the system will automatically compare the expression profile of podocyte versus the rest of kidney cells and show me some of these genes that can be used to distinguish podocyte from the rest. Okay, and now we come to the last and also I think the most used tool is the DGE. In the DGE, what you can do is just simple. You define the true population of cells based on the ontology or even the gene expression. You can add the gene expression into each of the cell population, and you can find the differentially expressed gene. One feature that I would like to highlight is the ability to view the expression of gene in individual sample and studies within the dataset, within each dataset in our database. So you can have a quick look on like whether the gene expression is uniform or is not. It's just like highly expressed in some specific studies. Okay. And yes, that's like some quick look of the features. In the upcoming session, we will have like more specific use case so that you can understand better about how these features can be used in action. And yes, so that's for the upcoming weeks. But for this week, I think that's everything that we have today. And now we would like to ask you some question about the new database so that your input could be very helpful for us. Okay, let me show you the poll. Okay, the poll is here. Please share with us, like, let's say, what kind of studies what kind of data that you would be most interested to see in Talk2Data so you can incorporate them. And also, what kind of ontology, what kind of metadata that you want to have. Because for now, we have 13, let me show you quickly. We have 13 here also. Let me just go here. Here are all of the 13 metadata information that we have. Please let us know which one is like the most useful for you, and what else? What else do you want to have in the database? And also, please share with us like what is the research area that you're working with, like either immunology, neuroscience, or cancer, so that our team, when we curate, when we find the studies, we can have a better view of like what is the current trend and what is the thing that we should collect the data for. Okay. Oh, we have one question from Wang Wuxing. Okay. Is this possible to share the inflammatory disease? Yes, of course. I can show you right away how can you create that one on the Talk2Data. So you can go to, okay, let me go back to the study search here. Right. So this is the default, the whole database, right? This is the whole database. Now I will try to show you. Yes. Okay. Yeah. Let me right here. Oh, we have a question from Maria also. Metabolic diseases. Oh, thank you so much, Maria. I will take note of this one and input it into our survey. Okay. Let me just quickly go here. Okay. So Wang is asking for the inflammatory disease. What can you do in here is you can go to the study search. You can open the filter in here and you can, under the condition, just type in inflammatory disease. Okay. I think my internet connection is unstable right now again, so it cannot load. But basically, that's how you can do. Just open the filter under the condition, type out inflammatory disease and apply them, select all to create an atlas. In here, I am selecting disease with both inflammatory disease and normal control in one study. So I want to make sure that, they all have control with them. So therefore, the number of studies are a little bit, a little bit lower at 154 studies. But yeah, that's the concept. Okay, now let me go back to the slide again. So we can have like okay. This one, answer. So please let us know if you have any question.
Yeah, as the polls are coming in, I'm seeing that we have a lot of interest in drug perturbation studies, definitely spatial transcriptomics, disease progression studies. as well as including the human phenotype ontology or HPO as a standardized ontology into the database as well.
I'm back.
Welcome back.
Oh my gosh, my connection. I'm really sorry. But yes, please let us know if you have any other questions. And also please answer the survey so that we can make, a better database. Okay. Yeah. Okay. Oh, I think we're a little bit over our time here. So, we still have one question. Oh, Charles. Oh, thank you, Charles. Oh, long time no see. Thank you so much for your kind words. And we're looking forward to see you again on our meeting. But yeah, thank you so much, Charles. Okay, so I think that's everything for today. And we will come back again later with, more use case on the new database and also the new Talk2Data. Please let us know if you have any question. Just, you can always send an email through support and we will always be there. And yes, see you next week.
Thank you everyone
Bye.
for joining today. Bye everyone.